yeast artificial chromosomes are, I can say that YACs. E. coli, it is not the single host that can be used to clone larger DNA fragments or the fragments from any source. इसके अलावा कुछ और भी ऑर्गेनिज्म हैं जिनको यूज़ किया जा सकता है टू क्लोन डीएनए फ्रैगमेंट्स फ्रॉम डिफरेंट सोर्सेस. तो इसकी जो एक एग्जांपल है वो है यीस्ट और स्कैरोमाइसिस. स्कैरोमाइसिस ई. कोलाई की तरह इसका भी जीनोम वेल स्टडीड है. इट इज़ वेरी सिंपल टू ग्रो इन द लेबोरेटरी. तो इसको भी यूज़ किया जा सकता है एज अ होस्ट ऑर्गेनिज्म to clone recombinant DNA. If we have a look again on the map of yeast artificial chromosome, then we will find that it contains different selectable markers that can be used for the selection of recombinants. Then it contains a portion which is called as the centromere. If you remember, centromere is a portion where the microtubules or the spindle fiber it attaches, and it play roles in the division of the chromosome during the mitotic process. Another portion of the yeast artificial chromosome that is important, its origin of replication. So the yeast artificial chromosome, it contains different DNA segments that are important. It also contains restriction sites for different type of enzymes that can be used to clone DNA fragments. So here, the yeast artificial chromosome, it is digested with suitable restriction enzyme like the foreign DNA that can be partially digested to create larger DNA fragments. And then these are ligated together, and then this recombinant artificial yeast chromosome, it can be inserted into the yeast cell. If you will remove the cell wall of the yeast, then it is very easy to infect it. And here you should keep in mind that if we want to clone larger DNA fragments for their separation, we can use pulsed gel electrophoresis. This pulsed gel electrophoresis, it is similar to gel electrophoresis, but it is used to separate larger DNA fragments in an agarose gel. So because of this reason, this pulsed field gel electrophoresis, it is important. Once the segments, they are purified, separated, then they can be used to ligate with yeast artificial chromosome. Next is the strategy that we can use for cloning foreign DNA into yeast artificial chromosome. As I told you that it contains its own origin of replication so that it can replicate independently from the host chromosome. Region for centromere so that it can separate during the mitotic division. Selectable markers, that may be anyone for any antibiotic, like selectable marker 1 or selectable marker X or Y, that may indicate any antibiotic, or any other selectable marker. And telomeric DNA, it is also there, and within this telomeric DNA, BAMH1 restriction sites are there. One can digest with BAMH1, so this fragment of DNA it is discarded. And then in the further step, you will get a linear yeast artificial chromosome that is flanked by a region of DNA which is called as telomeres. And in the next step, it is further digested with EcoR1 enzyme so that left and right arm, they will get separated. After separation, in the next step, when these left and right arms, they get separated, then larger fragments of foreign DNA that have been digested with EcoR1, these are the fragments of foreign DNA that are comparatively larger in size. So they are mixed together, and the foreign DNA it is inserted here, which is comparatively larger DNA fragments. When recombinant yeast artificial chromosome it is produced, then it can be used to infect yeast cell. And yeast cell can be further treated with different type of enzymes. So suppose this is a yeast cell, it can be treated with enzymes so that its cell wall it is digested and you can get a spheroplast. Spheroplast means that it is also a type of protoplast that is cell wall-less, and now it is easy to infect. And then after infection, it can be, recombinants can be selected on the basis of some selectable markers that may be X or Y marker. So this is one of the strategy that we can use for the cloning of larger DNA fragments, especially from eukaryotes like the humans, to clone by using yeast artificial chromosomes. Yeast artificial chromosome like the bacterial artificial chromosome, इनको भी यूज़ किया जा सकता है to clone somewhat larger DNA fragments. तो बैक्स के मुकाबले में जो यैक्स हैं, इनमें जो DNA क्लोन किया जाता है उसका साइज़ कंपैरेटिवली ज़्यादा होता है. जो मिनिमम साइज़ क्लोन किया जा सकता है, इट मे बी अप टू 150 बेस पेयर्स और 15 किलो बेस, एंड इट मे गो अप टू 2000 kilo bases. So comparatively larger DNA fragments that may be up to 2 into 10 raised power 6 base pairs, they can be cloned. So it is a very large DNA fragment, and especially it can be used to clone eukaryotic DNA like the human genome. If yeast artificial chromosome, it will contain the foreign DNA that is less than 100,000 base pairs or 100 kilo base, then it will be degraded after its transformation into the yeast cell. So smaller fragments, they are not stable, whereas the larger fragments that are above the size of 150 kilobases, they are more stable for cloning with yeast artificial chromosomes.